Hello, my name is Yiqing, a junior at Academic Magnet High School in North Charleston, South Carolina, and I'll be presenting my research on an in vitro model for cigarette smoke-induced invertible disc degeneration. I'll go through an introduction, followed by the aim of the study and hypothesis, then I'll explain the methods and results, and conclude with the key findings. First of all, why do we have low back pain? Low back pain is the second most common reason for hospital visits worldwide, and for many it becomes a constant companion. But what if the root cause isn't just a sprain or strain, but the gradual degeneration of the invertebral discs in your spine? Invertebral discs, IVD, are located between each vertebrae and are essential for spinal flexibility and load distribution. When the IVD degenerates, the core dehydrates, the outer layer weakens, the end plate calcifies, and the disc height decreases. This breakdown of the disc triggers the growth of blood vessels and nerves into the disc, leading to inflammation and chronic pain. There exist several risk factors for IVD degeneration, including smoking, genetics, diabetes, and alcohol consumption. Among these, smoking is a primary factor. Smokers are nearly 4 times more likely to develop disc herniation and have 20% higher degeneration scores than non-smokers. Additionally, they often experience delayed recovery after surgery. Despite known risks, cigarette smoking remains a major health issue. In the US, 11.6% of adults are cigarette smokers. In South Carolina, the prevalence is even higher at 15.4%. IVD degeneration has no cure, only pain management options until spinal fusion, surgery, or disc replacement becomes necessary as the condition worsens. Currently, no therapeutic strategies to combat IVD degeneration exist. Since it remains incurable, more research is needed to prevent progression. Additionally, smoking's impact on IVD degeneration compared to its role in cancer and respiratory diseases is understudied. This leads us to the main goal of this study. We aim to develop an IVD organ culture system with standardized preparation of cigarette smoke extract CSE to investigate the mechanisms behind cigarette smoke-induced IVD degeneration and explore potential early therapeutic strategies to restore IVD cell function. Specifically, the role of oxidative stress will be evaluated. Oxidative stress arises when the amount of reactive oxygen species exceeds the capacity of antioxidants to neutralize them, resulting in cellular and tissue damage. Cigarette smoke contains nicotine and other harmful chemicals which can increase ROS levels and cause oxidative stress in the cells. N-acetylcysteine, NAC, is an FDA-approved drug for treating Tylenol overdose. It works by reducing harmful ROS and increasing levels of glutathione GSH levels, a natural antioxidant in cells. This helps protect cells from oxidative damage and keeps them working properly. This brings us to our hypothesis that CSC-induced oxidative stress is a key driver of IVD degeneration. NAC as an antioxidant can mitigate oxidative stress and restore cell function, offering a potential therapeutic approach for CSC-induced IVD degeneration. Now I'll present the methods and results. Here's a quick overview of the experimental design. First, CSC was prepared and quality checked. Next, rat IVDs were isolated, cultures as explants assigned to different treatment groups, and then both intact and dissected regions were used for functional assays. I'll go into details in the upcoming slides. Let's begin with the preparation of CSC. Research-grade cigarettes were used for this process, and a syringe was used to draw 35 milliliters of smoke over 2 seconds. As the syringe was pulled, the mainstream smoke bubbled through the culture media in a falcon tube, mimicking the act of inhaling smoke. After each puff, it was followed by a 28-second rest or exhalation interval, and the smoke in the syringe was removed. Each cigarette was fully consumed after 14 to 15 puffs, and this procedure produced a 100% CSC solution, which was then used for the subsequent experiments. The quality of the CSC was assessed using 2 methods. First, light absorbance at 320 nanometers was measured to evaluate the amount of organic compounds present. Second, nitrite levels were measured using a grease reagent kit, which produces a pink azo dye, detectable at 548 nanometers. As shown in both graphs, the results were consistent across each preparation with no significant differences, confirming that CSC preparation is reliable and that the key compounds are at the appropriate levels. Now, let's discuss the IVD isolation, culture, and treatment. IVD were isolated from 6-month-old SD rats and cultured as explants to preserve their native tissue architecture. After a 2-day acclimation period, the discs were randomly assigned to one of 3 treatment groups. Control, 10% CSE, and 10% CSE with 5 millimolars of NaC. The 10% CSE exposure mimics the effect of smoking about 1 pack of cigarettes per day, representing heavy chronic smoking in humans. Following 3 days of treatment, the IVDs were either used intact or dissected into 3 distinct regions: the nuclear pulposus NP, the inner gel-like core, the annulus fibrosus AF, the outer fibrous layer, and the cartilaginous end plate CEP, the interface between the disc and the vertebrae. These are then analyzed for cell viability, oxidative stress, calcification, and apoptosis. To evaluate cell viability, a live dead assay was performed using calcine AM and ethidium homodimer. Calcine AM stains live cells green, while ethidium homodimer stains dead cells red, allowing for clear identification between the two cell populations. As shown here, the control group has a majority of cells in all 3 regions that exhibit green fluorescence. This indicates high cell viability and that our current IVD culture system is well maintained under the current conditions. On the other hand, the CSE treated group displayed a significant increase in red dead cells, indicating CSE-induced cell death. To test for oxidative stress, an ROS assay was conducted. For this assay, the fluorogenic probe DCFH deoxyQ was used, which generates fluorescent DCF when oxidized by ROS. The intensity of the fluorescence is directly proportional to the levels of ROS. In graph A, intact IVD was used for the ROS assay. CSE treatment resulted in a more than 2-fold increase in ROS levels, indicating significant oxidative stress in the cells. In graph B, the IVD were separated into 3 regions: the AF, CP, and MP regions. The same tests were conducted, and we found that oxidative stress varied across the regions. The CEP region was the most affected. The NP region is not shown in the graph, as its ROS levels were below the detection limit. In both graphs A and B, co-treatment with NaC significantly reduced ROS levels, indicating its potential to alleviate CSE-induced oxidative damage. In addition, a GSH assay was conducted to assess oxidative stress. As mentioned earlier, GSH is a key intracellular antioxidant. Higher GSH levels indicate lower oxidative stress, as it helps neutralize harmful ROS. Cellular GSH levels were measured using a thal fluorescent probe, which increases fluorescence 470-fold upon binding to GSH, allowing for precise quantification. The results showed an inverse relationship between ROS and GSH levels. In graph A, the CSE-treated group had the lowest GSH, while the NAC-treated group had the highest, highlighting NAC's protective effect against CSE-induced oxidative stress. In graph B, the NP group had the highest GSH levels, indicating its resistance to oxidative damage. Next, alkaline phosphatase ALP activity was assessed as a marker of hypertrophy, which plays a critical role in cartilage calcification. Samples were incubated with 4-nitrophenol phosphate, a substrate that is converted by ALP into yellow 4-nitrophenol. The resulting product was measured at 405 nanometers to quantify ALP activity levels. As shown in graph A, treatment with 10% CSE caused a 2-fold increase in ALP activity in the intact IVD. NAC co-treatment significantly reduced ALP activation. Moving to graph B, the highest ALP activity levels were observed in the CEP region, with values 4 times higher than the AF region and 8 times higher than the NP region. These findings highlight the CEP region as the primary site of ALP activation, which may contribute to early calcification and degeneration in this region. In addition, a caspase assay was performed to measure caspase-3 activity, a key marker of apoptosis. The assay detects caspase activation by measuring the release of fluorescence rhodamine upon the cleavage of the DVD peptide from the substrate. Similar to trends observed in the ROS and ALP activity studies, CSE treatment significantly increased caspase-3 activity in the intact disc, as well as individual regions, leading to apoptosis. Co-treatment with NAC reversed these effects, suggesting its potential protective role. In conclusion, an IVD organ culture system was developed to investigate the impact of CSE on IVD cell function. This study is the first to link CSE-induced oxidative stress with IVD apoptosis and calcification, highlighting the CEP as a key affected area. Since CEP calcification is a primary sign of disc degeneration, targeting CEP calcification could enable early diagnosis and intervention to slow or prevent disc degeneration. Restoring redox balance with NAC offers a promising new approach for IVD degeneration treatment. This study does have several limitations. First, the smaller sample size limits how widely the results can be applied, and since we focused on the direct effects of CSC on IVD degeneration, we didn't explore other factors like nutrient deficiencies. Additionally, the study lacks depth in understanding the cellular mechanisms involved. Lastly, while oxidative stress seems important, more research is needed to confirm its role as a therapeutic target. In the future, we will include more rats and those of both sexes to validate the results. We'll look at how nutrient deficiencies like low oxygen and glucose in different CSE doses affect the discs. We'll explore how oxidative stress causes damage at the cellular level, focusing on mitochondrial, endoplasmic reticulum dysfunction, and protein changes. And then we will also optimize NAC for the treatment of smoking-induced disc degeneration. Hopefully, completing this project will lead to large animal studies and small-scale clinical trials, helping develop a new treatment for early intervention in IVD degeneration. I sincerely thank Dr. Wu for providing the resources and opportunity at MESC, as well as his team Josh and Hui for their invaluable guidance. I would also like to thank my teacher at Academic Magnet, Ms. Roop, for her continuous support. And finally, I would like to thank IYRC for providing me the opportunity to present my research. Thank you very much.